Hello everyone. This presentation of Rhizopus. Rhizopus is a fungus. This is also made by a student. So you can see. And this presentation made by Drashna Ben. Now you can see your computer screen. Structure of Rhizopus. So occurrence. Rhizopus is a genus of common saprophytic fungi on plants and specialized parasites on animals. They are found in moist or damp places. They are found on organic substances like vegetables, fruits, bread, jellies, etc. Some Rhizopus species are opportunistic human pathogens that often cause fatal disease called mucormycosis. Classification: Kingdom fungi, Division Mycota, Subdivision Eumycotina, Class Zygomycetes, Order Mucorales, Family Mucoraceae, Genus Rhizopus. Thallus structure: They are fast-growing fungi and have a cottony appearance. The body of Rhizopus consists of branched mycelium. The mycelium is sinusoidal and composed of three types of hyphae: stolon, rhizoids, and sporangiophores. Stolon is the internodal region. It is aerial, forms an arc and touches the substratum, forming nodal region. Rhizoids are formed where the stolon touches the substratum at nodes. They are branched, anchor the mycelium to the substratum, and absorb food. This is the structure of Rhizopus. Sporangiophores are the aerial and reproductive mycelia. They are branched and consist of sporangiospores terminally. Sporangia are globose, often with a flattened base, grayish-black, powdery in appearance, up to 175 millimicron in diameter, and many spored. Structure of hyphae: The hyphal cell wall is microfibrillar and consists mainly of chitin and chitosan. Other substances like polysaccharides, proteins, lipids, etc. are also present. A cell membrane is present inside the cell wall, which surrounds the protoplast. The protoplast contains many cellular organelles, such as the mitochondria, endoplasmic reticulum, ribosomes, and vacuoles. Numerous nuclei are also present inside the protoplast. Droplets of oils and glycogen are the reserve food material found in the cytoplasm. Part of hyphae showing detailed structure as seen under electron microscope. Nutrition in Rhizopus. Rhizopus is a member of the fungi kingdom. It uses a heterotrophic feeding strategy. It is a saprophytic because it consumes the soil's dead and decaying materials. In the dead and decaying materials, the organic compounds are broken down into simpler compounds by the saprophytes. Amino acids are formed from proteins, fatty acids from lipids, glycerol from lipids, and glucose from starch. Endocytosis allows for the reabsorption of these substances into the hypha, where they are distributed throughout the mycelium complex and used for the plant growth and development. Reproduction in Rhizopus: It reproduces by vegetative, asexual, and sexual methods. Vegetative reproduction: Sometimes the thallus of the Rhizopus breaks accidentally or due to some other factors into the new fragments, after which each fragment gives rise to a new thallus on favorable conditions. Asexual reproduction: The asexual reproduction can take place through both sporangiospores and chlamydospores. First, the vegetative hypha grows to form sporangiophore that is long and slender. Then the sporangiofold develops and enlarges at the apical region to the form columella of various shapes and sizes. This columella gives rise to the large and round sporangium by pushing its cytoplasmic material to the peripheral wall. After that, differentiation of sporangia occurs that gives rise to the formation of spore sac in between the sporangia and columella. A spore sac carries the sporangiospores that are oval, unicellular, multinucleate and non-motile. These sporangiospores multiply their number within the spore sacs and the sporangial wall ruptures during the extreme conditions and facilitates the release of sporangiospores out of the wall. In the environment, the sporangiospores remain in a dormant or resting state. When favorable conditions return, these sporangiospores form germ tube or undergo germination to develop new vegetative hyphae. Asexual reproduction of Rhizopus by sporangiospores. During unfavorable conditions like insufficient food, material and water, the protoplasm is surrounded by a thick and nutrient-rich wall. This is called chlamydospore. This chlamydospore then detaches from the vegetative hyphae and remains in the resting phase. When this chlamydospore gets moisture, it forms a germ tube or undergoes germination process to develop a new thallus. Asexual reproduction of Rhizopus by chlamydospores. First, the plus and minus thallus come in the contact with each other. Then conjugation occurs between the positive and negative thalli through an outgrowth developed by positive and negative thalli. The septum develops between the progamidengium and eventually plasmogamy occurs. That results in the formation of cynogametes. Then karyogamy occurs, by which the gametes of both the plus and minus thallus fuse with each other to form a zygote. A zygote goes through the maturation phase and becomes encircled by a thick-walled structure called zygospore. There are two layers, namely an outer and inner layer that surrounds the zygospore. The zygospore lives in a resting phase for some time. On favorable conditions, zygospore forms a germ tube and germinates by forming a new vegetative body. Therefore, a Rhizopus reproduces through vegetative, asexual and sexual means to form differentiated hyphae, vegetative and reproductive hyphae. Sexual reproduction of Rhizopus by gametangial contact or conjugation. Two hyphae of differing mating type grow side by side and produce branches that grow towards each other. The tips of these branches develop into gametangia, a structure that produces gametes. The gametangia and then the gametes fuse. The resulting zygote develops into a resistant zygospore. Asexual and sexual reproduction, schematic diagram in front of you. Economic importance. Many Rhizopus species are commercially used to produce various chemicals and alcoholic products. Rhizopus oryzae is used in the production of cortisone and lactic acid. It is also used for bioabsorption of heavy metals in wastewater. Stoloniferin used to produce lactic acid, fumaric acid and cortisone. Delamar is used to produce biotin and fumaric acid. Tempeh, a traditional soybean fermented product of Asia, is prepared using the Rhizopus species. Many traditional alcoholic beverages is prepared using Rhizopus. Thank you very much. Hello everyone. This presentation of